The COVID-19 pandemic has made us all feel unsafe outside in the public. COVID-19 can be transmitted through surfaces, and when the world reopens and we enter the new normal, how can we know that door handles, railings, or the things we touch are safe? This issue of surface transmission is the origin of the idea for this research. My name is Ryan Ethan Yeo from SMP Citati Christian School West, and my research is titled Biosynthesis of Copper Nanoparticles, Cu-Ageratum conyzoides, Antimicrobial Agent for the New Normal. So, what are copper nanoparticles? They are nano-sized particles of copper, meaning the individual particles are very, very small. Copper is known for its antimicrobial, antibacterial, and antiviral capabilities. Copper nanoparticles' small size allows it to interact with bacteria cells or a virus, break down its outer defenses, and destroy their DNA or RNA. How does copper perform against SARS-CoV-2, the virus that causes COVID-19? Well, SARS-CoV-2 can remain infectious for 4 hours on copper surfaces compared to 24 hours on cardboard, 48 hours on stainless steel, and 72 hours on plastic. This shows the potential it has to be a weapon in this pandemic and prevent even more infections. Copper can be used in face masks, personal protective equipment, hospital equipment, door handles, and many other applications. Here are some examples of the research being conducted right now. First, Lippi developed a cloth disinfector mask using a copper coating. A team at Ben-Gurion University developed a surface coating material using copper nanoparticles. And lastly, a company called Copper 3D designed a 3D printed face mask also using copper nanoparticles and PLA recyclable plastic. Unfortunately, the usual method to produce copper nanoparticles is toxic and not eco-friendly. This is where the biosynthesis process comes in. It is relatively easier, eco-friendly, and cheaper than conventional chemical processes. Essentially, the copper salt precursor mixes with plant extract to produce the nanoparticle, from big crystals to small nanoparticles. The plant extract contains chemicals that break down the bitter copper salts into the copper nanoparticle. These chemicals are called biomolecules or phytochemicals like flavonoids, saponins, alkaloids, and tannins. Meanwhile, Ageratum conyzoides, also known as banutan, is an invasive weed also used as traditional medicine. It contains high concentrations of the biomolecules and has not been used to synthesize copper nanoparticles before. My objectives with this research are to first, synthesize copper nanoparticles with Ageratum conyzoides leaf extract, and second, research the results of synthesized copper nanoparticles, investigating their shape, size, and antimicrobial capability. I want to answer the questions first, how to synthesize copper nanoparticles, and then are there phytochemicals needed for biosynthesis present in Ageratum conyzoides leaf extract? How are the characteristics of the resulting copper nanoparticles? And lastly, how are the antimicrobial capabilities of the resulting copper nanoparticles? The plant experiment design comprises of extract preparation, phytochemical analysis, nanoparticle biosynthesis, characterization with SAM, and antimicrobial tests. Due to the pandemic and limited access to schools and labs, steps 2, 4, and 5 could not be conducted, so secondary data was used for the phytochemical analysis, characterization, and antimicrobial tests. The extract was prepared by first washing the fresh leaves to remove dirt, then drying, grounding into small pieces, and then the leaves were heated with water to produce the extract. The phytochemical analysis is conducted to determine the presence of active biomolecules needed for nanoparticle biosynthesis in the plant extract. The compounds tested are flavonoids using sodium hydroxide, alkaloids using Wagner's reagent, tannins using ferric chloride, and saponins, which can be identified by shaking the extract. For the biosynthesis process, there are specific variables and conditions that have been set. First, the independent variable is the ratio and concentration of extract to copper sulfate, which is 1/10, 1/3, and 1/2. For the control, pH was adjusted to 10 using sodium hydroxide, and the temperature was around 60 degrees Celsius. The dependent variables are the diameter and shape of the nanoparticles and the zone of inhibition for the antimicrobial test. In the actual biosynthesis process, copper sulfate solution is mixed with the extract in the different ratios, as I said before, while being stirred and heated. This creates 3 different samples that can be analyzed. Standard electron microscopy is used to see the size and shape of the nanoparticles. Next, we have the antimicrobial test using the disc diffusion method. Bacteria culture taken from hands and door handles are placed onto agar in the petri dish. Then, discs of filter paper are dipped into the nanoparticle solution and placed onto the agar. The nanoparticles then diffuse into the agar, inhibiting bacterial growth. This inhibition is measured by the width of the zone of inhibition around the filter paper. So, here are the results from the secondary data. Literary research was conducted using many journal papers to predict and show the results of the copper nanoparticles that were formed by biosynthesis. First, the phytochemical analysis showed that the aqueous extract of Cu-Ageratum conyzoides leaf extract did contain phytochemicals like alkaloids, tannins, and saponins that have an active function in the biosynthesis process. Only flavonoids were not present, and even so, the other chemicals indicate that the extract can effectively synthesize copper nanoparticles. Then we have the result of the biosynthesis process. In laboratory testing, there was a color change from green to a dark brown after an hour that indicates that the reaction was complete and copper nanoparticles were successfully formed. Here we have the results of characterization. Here are some examples of SEM micrographs, and the table shows the different sizes and shapes from the biosynthesis with many different types of plants. In general, the size varied from 10 to 250 nanometers with a spherical shape. For the antimicrobial test, the image in the table with the zones of inhibition show the significant antimicrobial activity of copper nanoparticles against human pathogens. The conclusion of this research is that copper nanoparticles can be synthesized in a more eco-friendly way for use to fight COVID-19 in a new normal. For future work, first, all testing would be conducted in a laboratory using real experimental data, which was not possible this time. Then, research and experiment with the potential applications like a surface coating or a face mask that can be developed even more. Let us take care of our environment, keep things clean, and stay safe. Thank you.